The inner ear has two distinct portions: the vestibular portion, concerned with balance, and the auditory portion, the cochlea, which is concerned with hearing. The vestibular portion is made up of 3 semicircular canals. Each canal has a swelling at one end, the ampulla, and 2 membranous sacs, the utricle and the saccule. These contain fluid and sensory cells, which move in response to head movements and travel to indicate the body's status. The vestibular system works in conjunction with the eyes and the receptor cells in the joints of the body to continuously maintain our balance. The cochlea appears as a coiled tube in the shape of a snail shell, with between 2½ and 2 3/4 turns. It is wrapped around the acoustic portion of the auditory nerve. If we were to unravel the cochlea, it would be about 35 to 40 millimeters long. It is split into 3 distinct sections, known as the scala vestibuli, scala media, and scala tympani. The scala vestibuli and the scala tympani contain perilymph fluid. The scala media contains endolymph fluid. The Reissner's membrane separates the scala vestibuli from the scala media, and the basilar membrane separates the scala media from the scala tympani. Along the basilar membrane are 2 types of sensory cells, or hair cells, called the outer and inner hair cells. These are arranged in their rows according to the frequency, or pitch, of sound they detect, and covered by the tectorial membrane. Each hair cell has tiny hair-like projections, called stereocilia, attached to them. When sound energy is transferred to the cochlea, the basilar membrane vibrates up and down, causing the outer hair cell stereocilia to shear on the tectorial membrane above. This shearing action causes the stereocilia to bend, which opens the ion gates, leading to chemical changes and a resulting electrical charge inside their cells. These charges make a neural impulse, which travels along the auditory nerve to the brain and which is interpreted as sound. This complex